So the first reminder is that I did give you an active learning assignment to do as a homework assignment. I just took a look at that this morning, and there's still quite a few people that have not done that. That's due today. Okay, so it's not too late to do it. It shouldn't take too long to do it because we went over it in class. So make sure you get that done. Okay, make sure you get that done today. Second thing is the Connect assignment for Chapter 12. I also checked on that, and there were quite a few people who had not yet finished it yet. I think over half of you had finished it. Okay, so that is due before you take the third exam, which right now is scheduled for Monday. Okay, but I'm wondering if I shouldn't postpone it. Since I was out 2 days last week, I'm wondering if I shouldn't postpone it a day, just to give us some time to do some review and to give you just some time to process things. You want to do that?
Yes.
Okay, are you all going to be able to be here on Wednesday?
Yes.
Okay, I checked the athletic schedule. It looked like there was a home basketball games Wednesday, but they're in the evening, so you shouldn't have to get out of class this early. Okay, let's do that. Let's do that. I'll have to go on to Canvas and change the date for the exam. Let's postpone it. And I see, it doesn't look like everybody's here today. I don't know where everybody is, but if you see someone who's not here, tell them that, I'll send an email out too, but just tell them that I'm gonna postpone it until Wednesday, okay? So, today we're gonna start the next section in Chapter 12. Now that we've seen the mechanism of local potentials or graded potentials and action potentials, now we're ready to see what happens next, okay? So, as Paul Harvey used to say, this is sort of the rest of the story, okay? So we've seen how a signal can get from the axon hillock to the end of the axon, where the synaptic knobs are located. That's where the neurotransmitters are released, okay? This is called synaptic transmission. So we're going to look at the mechanism of synaptic transmission today, which is really fairly simple. We're going to talk a little bit about neurotransmitters and how they work, and then we're going to look at how our brain and our spinal cord integrates information that's sent by neurons. Okay? So, a synapse is a junction at the end of a neuron where it stimulates another cell. Another cell may be another neuron, or it could be an effector, like a muscle fiber. And most synapses in all vertebrates, humans, birds, cats, dogs, all animals that have a backbone, most synapses are chemical synapses because the transmission from the first neuron to the next cell is by way of a chemical called a neurotransmitter. Okay? So this is a simple schematic diagram of one neuron stimulating a second neuron. Okay? So the junction or gap between the two cells is called the synapse. Okay? And the first neuron in that sequence is called the presynaptic neuron because it's before the synapse. The second one is called the postsynaptic neuron. So what's going to happen, as we'll see in just a little bit, the presynaptic neuron will be stimulated by a local potential, and if that local potential is large enough, if it exceeds threshold, then it will trigger an action potential, which will travel along the length of that axon. When the action potential gets to the synapse, that will trigger the release of a neurotransmitter. This neurotransmitter diffuses across that gap between the two cells. In a chemical synapse, the two cells do not actually touch each other. There's actually a very short gap. And so the neurotransmitters have to diffuse across that gap, which creates a slight delay. And then the neurotransmitters will set up a local potential in the postsynaptic neuron. That local potential will then travel down the dendrites and over the cell body to the axon hillock, where another action potential may be triggered. Depends if the local potential is big enough or not. Okay, so that's sort of an overview of what we're going to be going through today. Now, as it turns out, there's different kinds of chemical synapses, depending upon what part of the postsynaptic neuron is stimulated. Okay? So basically there's three types of synapses, and these have three different names. The most common type of synapse is an axodendritic synapse. It's this one right here on the upper left, okay? So in an axodendritic synapse, an axon terminal of the presynaptic neuron synapses on the dendrite of the postsynaptic neuron. That's the most common type of synapse. That's always the examples we always show when we're talking about how synapses work. Okay? But there's other possibilities. It's also possible for the presynaptic neuron to synapse on the soma, or the cell body of the postsynaptic neuron. That's called an axosomatic synapse, because it's a synapse between the axon and the soma, or cell body. Okay? Now, the axosomatic synapse, it most likely would work exactly the same as if the dendrite was being stimulated. So you can trigger a local potential in the cell body as well. It's probably easier to trigger a local potential on a dendrite though, because that's where you have the most ion channels that can be activated by the neurotransmitter. Okay? But there's also ion channels in the cell body, so you could trigger an action potential there just as well. Okay? Then there's a third type of synapse. This third type kind of produces a different effect. This third type of synapse is called an axo-axonic synapse. Okay, this is very different. It's not the usual type of synapse. In an axo-axonic synapse, the axon of a presynaptic neuron synapses directly onto the axon of a postsynaptic neuron. Okay. Now that's not the usual sequence by which things occur. And so this axo-axonic synapse, what it can do is this presynaptic neuron can modify the amount of neurotransmitter that's released by the postsynaptic neuron. Okay? So it can modulate the amount or modify the amount of neurotransmitter that's released. Okay? And it can do that in two different ways, depending on what neurotransmitter that the presynaptic neuron releases. It can either cause an increase in the amount of neurotransmitter that's released by the postsynaptic neuron, or it can decrease the amount of neurotransmitter, depending on what neurotransmitter is released by the presynaptic neuron. So this is one way that a presynaptic neuron can modify the amount of neurotransmitter and therefore modify the response produced by a postsynaptic neuron. So this is one way in which neural integration can occur. And it's even possible you could have an axodendritic synapse which is going to determine whether this postsynaptic neuron even produces an action potential at all or not. And then you could also have a second presynaptic neuron that produces an axonic synapse, and that can modify the amount of neurotransmitter that's produced. So it's kind of a way of fine-tuning neural stimulation in the nervous system. And this probably occurs mostly in the central nervous system, in the spinal cord and the brain, where neural integration occurs. Okay, we'll talk more about neural integration a little bit later. All right. Now, there's also such a thing as an electrical synapse, but electrical synapses do not occur in the nervous system of vertebrates. In vertebrates, the nervous system uses chemical synapses. However, there are electrical synapses that occur in other kinds of tissues, like cardiac muscle. Okay? The cardiac muscle fibers in the heart do form electrical synapses with adjacent cells. And that way, once the heart is stimulated, those electrical signals can be conducted very rapidly through the rest of the heart so that the heart can function as a unit instead of just a whole bunch of individual cells. Okay? So electrical synapses do occur in cardiac muscle. They can also occur between neuroglia, which is, neuroglia I guess is found in nervous tissue, but it's not the cells that produce the neural impulses. And it also can occur in a type of smooth muscle called single unit smooth muscle. But in the nervous system, which we're talking about right now, it only uses chemical synapses. So let's look at the mechanism of a chemical synapse. And I'm going to summarize this a little bit, but to simplify it, it's really not that complicated. Okay? So the first step in releasing neurotransmitter at a chemical synapse is you have to have an action potential traveling down the axon of the presynaptic neuron, which is the first neuron. So this line here represents an action potential moving down the axon. Remember, that's the result of activation of voltage-gated sodium channels, which then become inactivated, and then potassium channels open, which repolarizes the membrane, and that action potential then spreads down the axon in a non-decremental fashion. So in other words, it occurs continuously without decreasing in magnitude. Okay? So when the action potential gets to the end of the axonal branches, the very end of the branches are called synaptic knobs. Inside the synaptic knob, there are numerous vesicles, or membrane-bound vesicles, which contain a certain amount of neurotransmitter. Okay? So the neurotransmitter is stored in these vesicles. When the signal gets to that synaptic knob, it activates a different kind of ion channel. It's neither sodium channel nor potassium channel. Instead, in the synaptic knob, there are voltage-gated calcium channels. When voltage-gated calcium channels open, that allows calcium to diffuse into the synaptic knob, because calcium is in a higher concentration in the extracellular fluid than in the intracellular fluid in the synaptic knob. So when the action potential gets to the synaptic knob, that activates voltage-gated calcium channels. Those calcium channels open. That allows calcium to diffuse into the synaptic knob, just like this diagram shows. Okay? Calcium then causes the synaptic vesicles to fuse with the cell membrane of the synaptic knob. And you can see one right here, fusing with the cell membrane. When those vesicles fuse with the cell membrane, the vesicle opens up to the outside. And that allows the neurotransmitter to diffuse into the synaptic cleft. Synaptic cleft is this gap. And there's actually membrane-bound proteins, cell adhesion molecules that hold that maintain this space in the synaptic cleft. It holds the two neurons the correct distance apart from each other, so that there's a small gap, but that it's not too large, nor do the cells touch each other. So when the neurotransmitter is released, it diffuses across the synaptic cleft, and then it binds to ion channels that are located in the dendrite of the postsynaptic neuron. Or if it happens to be an axosomitic synapse, these ion channels might be on the soma of the neuron. Depends on what type of synapse you have. Okay. So let's take a closer look at these ion channels. Whoops. Okay. Well, these ion channels, basically, to make a long story short, they're ion channels that allow sodium to go across the membrane when the neurotransmitter binds to the ion channel. And that allows those ion channels to open up, and so sodium starts diffusing into the dendrite of the postsynaptic neuron, and that produces a local potential. Okay? If it's a sodium channel that opens, it'll cause a depolarizing local potential. But some neurotransmitters will bind to other kinds of ion channels, like chloride channels. If a chloride channel opens, then negatively charged chloride ions move in. That will cause a hyperpolarizing local potential. Okay, so the type of local potential produced depends on what neurotransmitter is released by the presynaptic neuron. Okay? Any questions? So let me just summarize quickly. Action potential moves down to the end of the axon. That activates voltage-gated calcium channels, which open. That allows calcium to diffuse in. Calcium causes the synaptic vesicles to fuse with the membrane. When they fuse with the membrane, they open up, and that releases the neurotransmitter. And that stimulates the postsynaptic neuron. So that, in a nutshell, is how a chemical synapse works. Okay? All right. Now, as long as that neurotransmitter is bound to those ion channels in the postsynaptic membrane, then those ion channels will continue to be activated, and sodium will continue to diffuse in. You'll continue to get local potentials occurring. Okay? So there's some situations where you don't want to keep on stimulating the postsynaptic neuron. Eventually you want to stop stimulating it. Okay? Once the desired response is produced, you need to stop stimulating the postsynaptic neuron. And so synaptic transmission can be terminated or ended in three different ways. Okay? The first is that the neurotransmitter could just diffuse away from the synaptic cleft. That's one way that synaptic transmission can stop. Second way is the neurotransmitter can be taken up by the presynaptic synaptic knob and then broken down by an enzyme inside the presynaptic neuron. Okay? So it can be taken up by the presynaptic neuron. It actually can be recycled, or it can be broken down, and then the subunits can be reassembled into new neurotransmitter molecules. Or there may be an enzyme embedded in the membrane of the postsynaptic neuron. Okay? One example of this is the enzyme that breaks down acetylcholine. Okay, that enzyme that breaks down acetylcholine is embedded in the cell membrane of the dendrites of the postsynaptic neuron. And so when the acetylcholine is floating around in that synaptic cleft, it can bind to that enzyme, and that enzyme will break it down. Okay? So those are three different ways that neurotransmitters can be removed from the synaptic cleft so that synaptic transmission doesn't just keep on occurring. All right, so I mentioned an enzyme that breaks down acetylcholine. Acetylcholine is one of the most commonly used neurotransmitters in the peripheral nervous system. And so I'm going to see how many of you have been reading ahead in here, okay? So I didn't mention the name of that on purpose because I want to see if you know the name of it. So what's the name of that enzyme that breaks down acetylcholine at the synaptic cleft? See if you can figure it out. Oh, by the way, the code for these set of questions is acetylcholine. Since we're starting a new section of chapter 12, we're starting a new set of questions. Oh, we have a lot of people gone. Where is everybody? Is it just because it's Friday?
Girls basketball's done.
Oh, are they? Okay. Okay. Shouldn't be too many of them, though, are there? Okay, are we ready to see the answers? Okay, most of you said acetylcholinesterase, and that's correct. That's the name of the enzyme that breaks down acetylcholine. Okay. Acetylcholine was the very first neurotransmitter that physiologists discovered. And probably the reason that it was discovered first is because that's the most common neurotransmitter that's used in the peripheral nervous system. It's also used in the brain, but there are other neurotransmitters that are used in the brain, like amino acids. There are some amino acids that act as neurotransmitters. So there actually have been over 100 neurotransmitters that have been identified by physiologists. And these different neurotransmitters, some of them some of the different ones are found in different organs in the body than others. Some of them, though, produce depolarizing local potentials by stimulating the opening of sodium channels. Others produce hyperpolarizing local potentials by stimulating the opening of other kinds of ion channels, like chloride channels. Chloride is negatively charged, so if you open up a chloride channel, chloride moves in to the postsynaptic neuron, and that's going to make the inside of the membrane more negative, which will hyperpolarize the cell. That will make it more difficult for an action potential to occur in the postsynaptic neuron because it moves the membrane potential further from threshold. So it'll make it less likely for an action potential. A depolarizing action potential, however, moves the membrane potential closer to threshold, so that'll make it more likely for an action potential to occur. Okay, Now, we've seen that there are different ways in which two or more neurons can form a synapse. By forming different synaptic connections, our central nervous system can process information. And in a way, it can kind of make a decision as to whether or not a response should be produced or not. It can also determine the type of response that occurs, okay? Whether it's just a single action potential that's produced or whether it's a whole burst of action potentials, okay? So, and by having more than one synapse on a postsynaptic neuron, you can get different effects because local potentials can be added together. So you may have two small local potentials that are a result of a stimulus. If they occur about the same time, they can add together to form a bigger local potential, which may be above threshold. So there's different ways of combining inputs to postsynaptic neurons to produce different kinds of responses. Some neurons actually have as many as 40,000 synaptic inputs. So that's one of the reasons why the brain can do so many amazing things. We can learn how to do things, we can store things in memory. It's all a result of all these synaptic connections between neurons. Now, the graded potentials that I've been talking about in the postsynaptic neuron are called postsynaptic potentials if they're produced by a neurotransmitter. And there's two kinds of postsynaptic potentials that can occur in postsynaptic neurons. The first is an excitatory postsynaptic potential, which is abbreviated as EPSP, for excitatory postsynaptic potential. That results from a depolarization of the membrane in the postsynaptic neuron. So that's going to bring the membrane potential closer to threshold. Okay? The other type of postsynaptic potential is called an inhibitory postsynaptic potential. An inhibitory postsynaptic potential results from a hyperpolarization. And that's usually caused by an opening of chloride channels, or less commonly, the opening of potassium channels. Now, potassium is positively charged, but it's in a higher concentration on the inside of cells. So if you activate a potassium channel, potassium moves out of the cell. And if you move positive charges out of the cell, it's going to leave the inside of the membrane more negative. So that will result in an inhibitory postsynaptic potential. Okay? That moves the membrane potential further from threshold. That's going to make it harder for the postsynaptic neuron to produce an action potential. Okay, let's talk about some of the more important neurotransmitters. Neurotransmitters are actually classified into 6 different types based on their chemistry. I'm not going to ask that you know all 6 of those different classes of neurotransmitters. Like I said, there's over 100 of them. And they're divided up into 6 classes. But I do want to mention some of the more important ones in the peripheral nervous system and the central nervous system. So in the central nervous system, the 2 most important neurotransmitters are actually amino acids. Amino acids can be released by neurons and used as neurotransmitters. So in the central nervous system, glutamate and aspartate, which are 2 amino acids, produce excitatory postsynaptic potentials. So those open up sodium channels. Then there's 2 other amino acids in the central nervous system. One is glycine and GABA. GABA is GABA-aminobutyric acid. It's another amino acid. And both of these produce inhibitory postsynaptic potentials. So they would work by either activating chloride channels or potassium channels. Now, in the peripheral nervous system, the most common neurotransmitter, and the only one that's used in the somatic division, which activates skeletal muscles, is acetylcholine. But in the autonomic nervous system, norepinephrine is sometimes used. Norepinephrine is used mainly in the sympathetic division of the autonomic nervous system. The parasympathetic division, though, also uses acetylcholine. So acetylcholine is most common in the peripheral nervous system. Okay, now we're going to take a closer look at a form of neural integration. One of the ways that neural integration can occur is by a process called summation. Summation basically involves the combining of two or more local potentials on a postsynaptic neuron. And by combining the effects of two or more local potentials, you can produce a different effect. Okay, the effect is usually the sum of all of the local potentials that are occurring on the postsynaptic neuron at about the same time. Okay? So, if the combined effect of the local potentials is excitatory, in other words, if most of the local potentials are depolarizations, then it may cause an excitatory postsynaptic potential to occur. But if the majority of the local potentials are hyperpolarizing, then that may result in an inhibitory postsynaptic potential. Okay, now, notice it says it's important to note that most neurons have ion leakage across the membrane that causes them to fire at a very slow rate all the time. So most neurons are firing at a very slow rate continuously. And so I think it's important to note it's not whether or not a neuron fires that determines whether, let's say, a skeletal muscle fiber contracts. It's the frequency of action potentials, actually. Action potentials rarely happen individually. They usually occur in bursts. And it's the frequency of the burst that's going to determine the effect. If you have a very high frequency burst of action potentials, that's going to stimulate the muscle fiber. If you have just a very low frequency, though, it's not going to produce enough excitatory postsynaptic potentials in the muscle fiber to activate a contraction. Okay. So the net effect of summation actually only changes the rate of firing, not whether or not the postsynaptic neuron fires at all. So, there's two different kinds of summation. There's temporal summation and there's spatial summation. In temporal summation can result from a single presynaptic neuron stimulating a postsynaptic neuron. So what happens in temporal summation? Temporal refers to time. So it's the stimulation of the postsynaptic neuron over time, but at a very fast rate. So let's say this presynaptic neuron right here produces, let's say, 3 different action potentials in rapid succession onto this postsynaptic neuron. that will produce 3 individual local potentials. But if they occur close enough together in time, then they'll be combined to produce a larger local potential. They're added together. That's what summation means, is to add. Okay? So in temporal summation, you have 2 or more action potentials produced in rapid succession, and the result of that is that they combine on the postsynaptic dendrite to produce an even larger local potential. Okay? Because the more local potentials that produce, the more sodium channels that open. The more sodium channels that open, the more sodium goes in, and the larger your overall local potential. Okay? Second type of summation is called spatial summation. In spatial summation, our postsynaptic neuron is being simultaneously stimulated by 2 or more presynaptic neurons. So in order to have spatial summation, you have to have more than 1 presynaptic neuron. Okay. So in spatial summation, each one of these 3 presynaptic neurons is producing a local potential on the postsynaptic neuron at the same time, the very same instant. So what happens then is each of these will produce a depolarizing local potential, which will spread down to the cell body. Before they get to the axon hillock, they'll combine together, sort of like 2 waves combining when they meet, and that will produce a larger local potential, which may be large enough to exceed threshold once it reaches the axon hillock, in which case it would produce action potential. Okay? So these are 2 ways by which multiple local potentials can be added together to produce a larger effect. And this larger effect doesn't necessarily always have to be depolarizing. You can have hyperpolarizing local potentials that get added together by either temporal or spatial summation, in which case you get an even larger hyperpolarization, which would make it well, what that's going to do is going to decrease the frequency of action potentials produced by the postsynaptic neuron. Now, another way that we can get neural integration is by forming synapses with whole groups of neurons. Okay? And by doing this, it can produce different kinds of effects. One way is called a diverging circuit. Okay? In a diverging circuit, you have one neuron which has several axon branches. Since the axon branches, each one of those branches can synapse with different postsynaptic neurons. Then each of these second order neurons, they also can have more than 1 axon branches. And each one of those axon branches can stimulate another postsynaptic neuron. So in a diverging neural circuit, you start out with a single neuron, which theoretically could activate a whole population of neurons. So with a diverging circuit, you can have just a single stimulus activating a whole large group of neurons. Okay? There's actually a diverging circuit that's found in our brainstem. It's called the reticular activating system. This reticular activating system is made up of a large number of neurons that alert our higher brain centers. And so our reticular activating system is what causes us to wake up in the morning. It alerts our higher brain centers so we wake up. Okay? And the way it works is that in our brainstem, there's a diverging network of neurons. And you know, when your alarm clock goes off in the morning, that stimulates neurons in your ear that sends signals to your brain, and that activates a diverging pattern of neurons, which alerts our whole higher part of our brain, which causes us to wake up. Okay? Opposite of a diverging circuit is a converging circuit. It's just the opposite of a diverging circuit. So in a converging circuit, you have a large number of neurons, and two or more of these neurons synapse just on a single neuron. And so as you go through a converging circuit, the number of neurons that's activated gets smaller and smaller. So this converging circuit can be a way of our brain processing information, because by having two or more presynaptic neurons converging on a single neuron, the effects of each of those individual presynaptic neurons are combined together. And so that can allow our brain to integrate information. Okay? It may be that our brain is being exposed to a large number of very small signals resulting from very small stimuli. But if you have enough of those small stimuli, it may be important enough to warrant a response. And so you can combine the effects of all those stimuli to trigger an action potential in just a single circuit. Okay. Another type of neural circuit is called a reverberating circuit. So in a reverberating circuit, you have a neuron at the beginning that stimulates a whole series of neurons. But one of the neurons near the end of the series has a branch that feeds back to the beginning of the circuit. So what happens is, once you activate this first neuron, it stimulates the second, and third, and fourth neuron, which will produce output. But the signal will also feed back in stimulating the first neuron again. And so after the first output, these will get activated again, and it'll produce another signal. So this can produce a whole burst of signals. And that burst of signals may be long enough to produce the desired effect. There's some situations where you need to have a muscle contract for longer periods of time. And by stimulating that muscle with a reverberating circuit, it can cause a long enough burst of action potentials to produce the desired outcome. One example of this is the circuits in our brain which control our respiratory muscles. In order for us to breathe, our inspiratory muscles have to be stimulated for about 2 seconds, because that's how long it takes us to take in air in our lungs. And so those inspiratory muscles are controlled by reverberating circuits in our brainstem that produce about a 2-second burst of action potentials. And that burst of action potentials will occur until this reverberating circuit is inhibited by an inhibitory neuron, or it will just keep occurring until these neurons get fatigued. Once they get fatigued, they'll stop firing. Okay. In the case of our respiratory system, it's probably an outside inhibitory neuron that shuts it off. Okay. And then another example is a parallel after discharge. Parallel after discharge can also produce a whole burst of action potentials, but that whole burst of action potentials can't continue for as long, because there's no feedback loop in a parallel after discharge. So the way this works is you have a whole series of pathways in parallel rather than a series. You have them in parallel. one parallel pathway may only have one synapse. The next may have 2 or 3 synapses, and the third may have even more than that. So each time you have synaptic transmission at these synapses, that creates a slight delay, because it takes a while for the neurotransmitter to diffuse across the synaptic cleft. And it takes a little while for the ion channels in the dendrites to open up. So the more synapses you have, the more delay there is in the time it takes to stimulate this last neuron. So in this top pathway, since there's only one synapse, it's not going to take as long for that signal to get to this last neuron. So that will cause the final neuron to fire first. Then the second pathway, which has more than one synapse, it will also stimulate that final neuron, but it'll be delayed a little bit. It's going to take a little longer for it to get there. And the same thing for this third one. This third one would stimulate the final neuron last, because it has a lot more synapses. It's going to take longer for that signal to get there. So this will cause a burst of signals, but once all the parallel circuits have fired, then that burst of action potential is going to stop. So just another way of getting a burst of action potentials. So in our brain, we have trillions of neurons, and each neuron can form 40,000 synapses. So it's not surprising that our brain can do so many amazing things. In fact, the brain physiologist at one time said that the brain is so complex, they didn't think that we would ever be able to understand how it works, because it's so complicated. But actually, physiologists are starting to figure out some of the circuits that control other parts of our body. And they're using various molecular techniques to do it. Like they can identify different types of neurons according to the types of proteins that they have in them. And they can selectively stain them with a dye that's attached to an antibody that's selectively attached to certain proteins in neurons in the brain. And so they can identify different populations of neurons by doing that. So they're actually making a lot of progress. I don't think they'll ever figure out everything about how the brain works, but they are figuring out some of the simpler pathways. Okay. All right, well, let's see. So like I said, reality, one neuron in the brain may have as many 40,000 synapses. Each of these little things is a synapse. And so it's no wonder we can do so many amazing things. So, okay, let's do one more review question and we'll stop here because we're about out of time. We do have about a minute. So what will happen if a postsynaptic neuron is stimulated simultaneously by an excitatory postsynaptic potential and an inhibitory postsynaptic potential of the same magnitude? What would happen? So if you have one local potential that's excitatory and one that's inhibitory, and you get summation of those two together, they're going to cancel each other out, right? So nothing would happen. You would not get any action potentials. But if you have 50 excitatory action potentials and only 10 inhibitory ones, then you're probably going to get an excitatory postsynaptic potential. Okay. Whoops. So the correct answer is number 3. All right, we'll stop here. And so Monday we'll do some review, which I think will help.